Have you ever wondered how you become you? Your DNA holds the blueprint for your entire body, every single part of it. But how does a simple string of code actually build your eyes or your hair or the tiny machines that keep you alive? Today, we are going to follow that instruction manual, page by page, all the way from the blueprint to the finished building.
Okay, so here's the big puzzle right up front. Think of your DNA as the master blueprint for an unbelievably complex skyscraper. It is priceless, so it's kept locked away for safekeeping inside the cell's main vault, a place called the nucleus. But here's the catch. The construction site, where all the real building happens, is way outside the vault, in an area called the cytoplasm. So how on earth do the instructions get from inside the locked vault all the way out to the busy construction site?
And this image right here, this is basically our roadmap for the whole journey. We're going to start with that famous DNA double helix, see how its instructions get copied into a special message, and then watch as that message gets read step by step to build a chain of amino acids, which, believe it or not, is the backbone of every single protein in your body. But before we can follow the message, we have to understand the language it's written in. I mean, it's a code, right? And like any secret code, it's got its own alphabet and its own set of rules that we need to crack first. Now here's where it gets really mind-bending. The language of DNA is incredibly simple. Its alphabet has just 4 letters, A, T, C, and G. That's it. But the language of proteins, the things DNA builds, is way more complex. It uses 20, that's 20, different building blocks called amino acids. This creates a really fascinating puzzle.
So how in the world does that work? How can life write a recipe book that calls for 20 different ingredients when it only has a 4-letter alphabet to work with? Let's you and I just think through this logically for a second. Okay, what if we tried a 1-to-1 system? You know, 1 letter of DNA for 1 amino acid. A could be 1 ingredient, T could be another, and so on. Well, that only gives us 4 possible codes. We need 20, so we're still missing instructions for 16 other ingredients. That's not going to work.
All right, plan B. What if we use 2-letter words? Like AT or AG, CC. Well, that gives us 4 times 4, which is 16 possible combinations. We're getting so much closer. But we're still missing 4. We still can't specify all 20 of the ingredients we need.
And here it is. This is the absolutely elegant solution that life figured out. If we use 3-letter words, the number of possible combinations just explodes. It's 4 times 4 times 4, which gives us 64 possible words. And that is more than enough to give unique code to all 20 amino acids, with a ton of words left over.
And this 3-letter genetic word actually has a name. It's called a codon. So that's the secret. The genetic code isn't read 1 letter at a time. It's read in these 3-letter chunks, these codons. And each codon points to one specific amino acid ingredient for our protein recipe.
And this right here, this is basically the universal decoder ring for all of life. It's amazing. Scientists have mapped out every single one of the 64 codons and what they mean. You can literally look up any 3-letter sequence, like say TTC, and you'll see it codes for an amino acid called phenylalanine. This is the language for pretty much all life on this planet.
But get this, not all the codons are ingredients. Some of those 64 combinations act more like punctuation, or maybe traffic signals. There's one special codon, ATG, that's like the capital letter at the start of a sentence. It tells the cell's machinery, hey, begin reading the recipe right here. And then there are 3 different stop codons that act like a period at the end, saying, all right, the recipe is finished. You can stop reading now.
Okay, so we've cracked the code. We know the language. Now, let's get back to our first problem. How do we get this coded message out of that secure vault, the nucleus, and over to the construction sites? Well, this first step is a process called transcription. This problem of physical separation is something you'll see in basically every genetics textbook. The instructions are in one place, the nucleus, but the factory is somewhere else entirely, the cytoplasm. There has to be a go-between. We need a messenger that can safely carry those precious instructions across the gap. So, allow me to introduce our messenger, a molecule called RNA. It's a super close cousin to DNA, but it has one really important difference. It's usually single-stranded. So think of it like this. If your DNA is the gigantic, irreplaceable master reference book and a library that can never be checked out, RNA is the quick photocopy you make of just one page, a single recipe that you can actually take with you to the kitchen. So the process of making that photocopy, transcription, it happens in 3 pretty simple steps. First up is initiation, where the cell's machinery scans the DNA and finds that start codon. Then comes elongation, where it moves along the gene, reading it and building a matching copy, called messenger RNA, or mRNA for short. And finally, there's termination. The machinery hits a stop codon, and the finished mRNA photocopy is released, ready to make its journey.
So now our messenger, the mRNA, has the instructions, and it's leaving the nucleus. It's on its way to the construction site out in the cytoplasm for the grand finale. This is the crucial last step, translation. This is where the coded message finally gets turned into a real physical product.
Now to build our protein, we're going to need a whole kitchen crew. First, we have our recipe. That's the mRNA we just made. Then we have the chef, a molecular machine called a ribosome, which is basically the protein factory that's going to read the recipe. And finally, we have the chef's assistants. These are tiny molecules called transfer RNA, or tRNA. Each little tRNA is a specialist. Its only job is to go and grab one specific amino acid ingredient.
And here is where the magic happens. The ribosome, our chef, clamps onto the mRNA recipe and starts chugging along, reading it one codon at a time. For every three-letter word it reads, the exact right tRNA assistant shows up, carrying the matching amino acid. The ribosome then takes that amino acid and links it to the one before it, building a growing chain, kind of like stringing beads on a necklace. It just keeps going, codon after codon, until, bam, it hits that stop signal. The second that stop signal is read, the now finished chain of amino acids is released. It immediately folds itself into a very specific, complex 3D shape. And just like that, it becomes a working protein ready to do its job in your body. And there you have it. That is the incredible link from an abstract code to the physical you.
And so we've come full circle. We started with a secret code, written in just 4 letters locked away in the nucleus. Through transcription, a messenger copy was made. And then, through translation, that message was read by a ribosome to build a very specific protein. That, in the end, builds and runs your body. It's what makes you, well, you.
What's truly wild is that this incredibly precise process is happening in your body right now, trillions upon trillions of times every single second. It is the absolute foundation of life as we know it, which of course leaves us with one last fascinating question. With all this complexity, what happens when there's a typo? What happens when just one single letter in that vital genetic code goes wrong?